Hello, I am Mahmud, a student of Master of Research from Hawkesbury Institute for the Environment. Today I am going to talk about my project. This is broadly based on soil science and ecosystem ecology. And my project title is Quantifying Soil Organic Carbon Storage at Cumberland Plains Supersoil. And my supervisor is Professor Ellis Pangham from Western Sydney University. And this presentation will be in two parts. First part, I'll talk about the discipline and why carbon is important. And in the second part, I'll discuss about my project and key findings. Let's start. So what is an ecosystem? An ecosystem consists of all the organisms and the biotic pools with which they interact. An ecosystem processes are the transfers of energy and materials from one pool to another. Ecosystem analysis seeks to understand the factors that regulate the pools, which means quantities, and fluxes, which means flows of materials and energy through ecological system. As you can see in the picture, pools are in boxes and fluxes are in arrows. These materials include carbon, water, nitrogen, and human-added materials to the environment. These materials are found in biotic, which means non-biological pools, such as soils, rocks, water, and the atmosphere, and in biotic pools, such as plants, animals, soil, microorganisms. Next topic is soil and its composition. As my project is on soil organic carbon, so it is important to me. So I will discuss soil at the moment. So soil is a dynamic natural body on the surface of the earth, in which plants grow and is composed of minerals and organic materials and living forms. It is different from the parent material below in morphology, physical properties and constituents, chemical properties, composition, and biological characteristics. The soil consists of four major components, and these components cannot be separated easily because they are very intimately mixed with each other. In the pie chart in the slide, you can see organic matter is only 5%, but it plays a very critical role for the agriculture, ecosystem, and environment. My project will look at this organic matter, especially organic carbon, which we will discuss in the second part. Now, soil organic matter. It comes from living organisms. It has a very critical and complex composition. It helps to sustain other plant, animal, human microorganisms, also production and growth of agricultural products and other life on earth. Soil organic carbon is a part of soil organic matter. Living organisms in and above the soil converts carbon dioxide into sugar and eventually transform into various organic compounds and structures. When they die, it goes into the soil and with the help of a range of fauna and microorganisms, it decomposes and provides fuel for the energy. When organisms respire it to use the energy, it becomes into carbon dioxide. Soil total carbon is composed of two fractions, namely organic carbon and inorganic carbon. This organic carbon and its associated compounds exhibits a range of physical, chemical, and biological property of soil and that ultimately supports and backbone of all living things on the planet. Now, this is a cycle of carbon, but if we produce more carbon dioxide in addition to the natural amount, so the carbon dioxide concentration will be high in the atmosphere. It will keep more heat into the atmosphere, causing a global warming. This will exhibit more heat waves, expansion of desert area, natural fires in forest lands, melting of ice caps, shorter and warmer winter, and longer and shorter summer. Changes in pattern of rainfall, rise in sea level, flooding, and submergence of low-lying coastal areas. It has been understood that transformation in land use leads to soil organic carbon loss from soil profiles. Thus, finding a method to store more organic carbon into the soil will also help to fight climate change. Next slide, we'll talk about more about the soil global carbon cycle and its pools. Now the global carbon cycle. In the diagram, the numbers in red are the carbon fluxes provided by the human activity. The terrestrial biosphere contains the largest biological reservoir of carbon. There is nearly as much carbon in terrestrial vegetation as in the atmosphere, with 2 to 3 times more organic carbon in soils than in the atmosphere. However, human activities are now a significant component of the global carbon cycle. Human carbon emissions from combustion of fossil fuels increased 40% from 1990 to 2008, according to Canadell et al. 2007. Land use conversion releases an additional 1.5 petagram of carbon a year by biomass burning and enhanced decomposition. Together, these anthropogenic fluxes are about 15% of the carbon cycle by terrestrial or by marine production. Moreover, human carbon fluxes represent net additions to the atmosphere. So far, I have discussed the background and importance of soil organic carbon. In the second part, I will discuss about my project. My project is based on the soil of the Cumberland Plain superset. It is a site of the Terrestrial Ecosystem Research Network, commonly known as TERN, which enables ecosystem scientists, both nationally and internationally, a common platform for research and data sharing. This facility is situated next to the Western Sydney University's Hawkesbury campus. This Cumberland Plain woodland is a critically endangered ecological community. It faces major pressure, including invasive weeds, altered fire regimes, urban development, conversion to agriculture, and extreme climate events. In regard to my project, the use of land of the supersite is been identified into 3 categories. These are grassland. In the map, it is indicated with black lines. Another division is woodland. In the map, it is marked by the yellow line. And last type is marsh or bog is surrounded by a purple line. Now, objectives of my project. At the moment, there are 3 research questions. To achieve these objectives, a set of experiments was done. The most important one, chemical data, was provided by the supervisor. I will discuss about the method and results in the last. Now, talk about the soil sampling. Samples were collected in early 2015 during the initial University of Western Sydney, Hawkesbury Institute of Environment soil mapping project from 0 to 10 centimeter and 20 to 30 centimeter depth. In the map, yellow dots are the point from where soil samples were taken. As the samples are from 2 depths, so each point had 2 sets of samples. To determine Organic carbon and other materials. These samples were characterized for various chemical and physical properties following a range of methods. I'm going through these methods very briefly. All collected soil samples are dried in the oven at 40 degrees Celsius overnight and then crushed and then sieved in less than 2 millimeter. For the measurement of plant available phosphorus and potassium, it is typically done using Caldwell method. And to determine soil organic carbon, for the purpose of this study, the weight method described by Wackley Black is used. For the third objective of this study, to get the reading from the NIL machine, first samples were made into fine powder using a Resch mixture mill shown in the picture. Model is MM400 is used. Each sample was milled for 2 minutes at a frequency of 30 Hz per minute. For milling, two 35 millimeter milling cup lined with zirconium oxide and a ball of 15 millimeter zirconium oxide was used. These samples are analyzed using the NIR with a FOSS XGS rapid content analyzer over a range of wavelength from 700 to 2500 nanometers. Now, key findings. After having chemically analyzed, these results have been statistically analyzed using Microsoft Excel, and some graphs have been produced. The result showing in the screen, we found that marsh has the highest amount of organic carbon storage as expected, while grassland has a relatively low amount of soil organic carbon. Also, the available nitrogen content, which is a combination of nitrate and ammonia, is high in marsh but poor in woodland and grassland. Similarly, the histogram of sand percentage is done using the sand data on the type of land use. This exhibits that grassland has high content of sand than other two land types. In other analysis, the clay content found in the marsh is high, but grassland has lowest quantity. Moreover, the regression analysis was done to find out any relationship of various parameters in the study area. This regression showing a positive relationship in soil organic carbon percentage and clay percentage. Similarly, clay percentage and nitrogen content has a good relation as R squared value is more than 50% and data are around the line of best fit. Also, there is a good relationship between organic carbon and nitrogen as they have a high R squared value both in grassland and marsh. We also found a significant positive relationship between organic carbon and phosphorus in marsh and grassland at Cumberland Plain supersite. To discuss, we found marsh has the highest storage of soil organic carbon, probably because it is low-lying land and the nearby organic matter washed into it. This also supported by Chapin et al. 2011 on the topographically low position of Mars. Similarly, available nitrogen content is also high in marsh. High soil organic carbon allows the high available nitrogen. The other statistics of this study also suggest that this variation in organic carbon and nitrogen availability is proportional to the amount of soil particle content. These data also support that high clay content and low sand content leads to high soil organic carbon and available nitrogen. These claims also supported by the regression analysis. The clay content on organic carbon or nitrogen has a very good relationship. This is again because organic carbon is high in clay soil and organic matter is the source of nutrient release. For the third objective of this project, we have done some initial testing of samples using near-infrared analyzer. It requires more testing of the samples, calibration, and analyzing the data to get the clear link between the objective and the actual test data. To conclude,
Understanding of soil organic carbon content and other parameters are useful for the soil and environmental management. The expected outcome of this study will need more laboratory work, more soil sampling to ensure the results and generation of report. The future plan of this project is to look at the role of the microbial activity to hold more carbon in the soil and to improve nutrient availability. This will lead to the productivity of agriculture, ecological restoration, and services to the community. Also, the spatial technique can be used to predict nearby soils and broad picture of the soil resources around Australia.
I hope that you enjoyed my presentation. Goodbye.